At Deliflor, we are constantly seeking to improve our processes and products, where technology is a fundamental ally on our path of growth. We introduce to you the molecular markers, a technology that allows us to select and develop our varieties in a more scientific manner. This is how molecular markers work. Like all living organisms on our planet, our varieties are formed through the genetic information stored in the DNA. The offspring contains half of the genetic information of both parents in a relatively uncontrolled way. The chromosomes which we find in every cell of our body are built of this DNA. The DNA is built of 4 different construction blocks, bases A, T, G, and C, which form units of 2 complementary bases. The order of these bases will determine the development and behavior of the organism, as well as its inheritance and variations. This is what we call the genotype. For example, a chrysanthemum variety is genetically configured to have pink petal color, but this genetics, interacting with environmental factors, can modify the color of these petals. This interaction is known as the phenotype. We conduct multiple phenotypic tests to determine the viability of a variety. As there is an interaction with the environment, these tests can last for many years while we select the candidate that meets all the requirements. Aesthetic characteristics are not the only factors that can define the genotype. Perhaps even more important is the response a flower can have towards certain pathogens. This is where molecular markers come into play. Today, in our laboratory, we can test markers that characterize resistance to pests and diseases. To determine if a variety possesses a particular molecular marker, its genotype has great possibilities to exhibit certain characteristics, which can be demonstrated through a genetic test. For this purpose, we use qPCR. qPCR, or quantitative real-time polymerase chain reaction, is highly accurate. It shows in real time the amplification of a certain piece of DNA. In this sequence, the nucleic bases that make up the DNA are visualized as letters. Our goal is to search for a very specific series of nitrogenous bases within this vast sequence. The presence of this sequence allows us to identify the genetic characteristics of a variety. For example, for a chrysanthemum with a genotype resistant to fungus X, its DNA sequence might be ATCGA. But if the sequence is ATTGA instead, it indicates that this variety is susceptible. The resistant variety has a fluorescent label called VIC, while the susceptible variety has the fluorescent label FAM. What does this mean? It means that we can select in advance the varieties that meet the criteria we are looking for, discarding all the potential candidates that we would have otherwise grown and subjected to phenotypic tests for more than 3 years. This way, we can find that successful variety among 10,000 candidates in a much lesser time. This waste of resources, time, and contamination are avoided with this process. In the seedling phase, we are testing approximately 2,400 samples per month, and for clones, they are slightly less frequent. We also conduct pathogen tests, which is a significant activity in our laboratory. Last year, we had a total of 5,600 samples. This year, we have already reached 7,300 samples and counting. We can analyze different types of pathogens, such as viruses, viroids, bacteria, and fungi. In the fungal test, we use an enrichment step. We incubate a fungus for 7 to 14 days and isolate its DNA for use in qPCR. We are searching for more markers that can provide breeders with information on resistance, tinting capability, vase life, etc. These tests allow us to approach new and more resistant generations of flowers in a faster and more scientific manner. Science, in the chaotic world of genetics, sets us apart from being mere crossers. These and other processes are carried out in Deliflor's laboratory.